Okay, good morning, class, or good day rather. This is a recorded video for the exercises 4 and 5. So, before I will give you the methods for the exercise 4 and 5, I will just give you an overview about the lecture on protein biochemistry. Okay, so the exercise, to give you a glimpse of what is all about the exercise 4 and 5. So I have given you this in advance because it will be an experiment at home activity or exercise. So you are going to conduct this, and pwede din na you can conduct this simultaneously in one setting because this is just a simple method. I modified this method in order that the materials are available in your homes, and the of course the materials are just affordable, and you can just buy it from the stores. Okay, so that's the thing about the exercises 4 and 5. Yeah, 4 and 5. Okay, so before I formally discuss with you the protein biochemistry, so I would like to ask you this question rather. Sorry. Okay, so you have to describe your midterm exam specifically in InBt121 lecture in lab in the line of a song. So I expect you to, as long as you watch this, it is a requirement for all of you that you have to send a response by singing the song uh, of sing the song of your response for the midterm examination. Did you find it difficult or did you find it easy? You have to describe it in a line of a song and you have to record it. So just send a recorded message in the FB Messenger. So I am sure that you are familiar that there is a button there that you can record your voice. So just touch and hold, long press it so that you can start record and then sing a line of a song. Okay, so I am expecting you to have a response to this because I will kanang ako dyan mo panuktan class. So maybe I can discover that many of you have good voice, okay, have a potential of being a singer. Wow. So kana. So I'd like to know the feedback on your midterm examination. So that's it. So just message me how I have to require all of you to respond on the FB Messenger. Okay? So to continue, this is protein biochemistry. So first is the protein structure. So here in this picture, this is the illustration of the concept of the central dogma of molecular biology. I know that you are quite familiar with this one because you have encountered this many times in BTEC 21 rather. It was introduced to you, so you have to memorize it by heart because that's the central dogma of a biotechnologist. Yeah, you have to memorize it and fully understand the concept of this dogma of molecular biology. So it is first the replication, the DNA replication, in which the cell will undergo cell cycle, like for the eukaryotic cycle and the prokaryotic cells. Rather, it will undergo a cell division. So the DNA will then be replicated in order to maintain the fidelity of the information of DNA. So you know what the DNA is, the deoxyribonucleic acids. So it contains the information, the DNA sequences, the codes for the proteins of the organisms or the microorganisms or the plant cells, right? So it is really important that the cells that are divided, that cells that contains the DNA are in the same sequences are really uh, copied religiously, because if not, there will be mutations that occur, and there will be problems in the uh, transcriptions and translation in the production of the protein. So after that, there will be also the production of the proteins, in which it will undergo transcription, in which a production of mRNA. So there will be a copy messenger of there will be a copy of the DNA sequences, the complementary sequences, in which the messenger RNA is produced in that. So it will then be transcribed. So there will be a messenger RNA that is produced rather. And what's the difference of DNA and RNA is that it is the RNA is single-stranded, obviously, and also the DNA is a double-stranded, and also the sequence in the RNA has a is substituted by a uracil, right? So that's a uracil, UA. So not for the complementary base pairing in the RNA is adenine and the uracil, while the DNA is adenine and thymine. Okay, so that's the gist of the RNA transcription. So it follows a mechanism, and after that, there will be it depends if your messenger RNA is eukaryotic, because there will be mRNA splicing and modification, and after that, while in the prokaryotic cells or the bacterial cells, well, there'll be no modification or mRNA splicing. Okay, after this transcription, there will then be translation that occur, and the messenger RNA will then be transported in the ribosomes, in which this is the location that the amino acids are produced. These sequences from the RNA are then translated. So three sequences corresponds to a one codon that represents one amino acid. So in one amino acids, there are many different codons that will be represented. That's the degeneracy of the genetic code. So again, I tell you that there is in one amino acid, there will be 3 to 4 equivalent of the three sequences in the DNA, the RNA rather. Okay. After that, if there is now a translation is already done, so there will be post-translational modification if it is uh, if the protein structure is if it has a secondary or tertiary structure, so it will undergo post-translational modification, but it depends if that protein is from eukaryotic cells or prokaryotic cells. But definitely, it's more complicated in the eukaryotic cells. Okay, so and it also depends upon the function of the protein. Okay, that's how the central dogma of molecular biology is. Okay, that's just an overview, but not really the detailed one. So it's just a summary. Okay, so here, so as you can see that in the translation, as I go back, there will be production of amino acids that will chain of amino acids that form a peptide or protein. So the amino acids, by definition, is the building blocks of proteins, and there are 20 amino acids that are existing, but there are already recent reports that there are many amino acids that has been discovered, like in the other planets. Um, I just knew it when I was doing my MS. So that's how there are many newly discovered uh, amino acids, but not yet not included in the original existing amino acids because it might need further investigation for that. And then after that, the linear chain of amino acids from that forms a peptide or protein. So when can we say that it is a peptide, and when can we say that it is protein? So later we will know that how can we say it is a peptide, or how can we say this is a protein. So the amino acids is composed of many elements or chemicals. No, so one is the central carbon. It is there is a central carbon there, and then there is also a chiral carbon mirror images, or the L and D isomers. But there are only mostly L isomers are just found in proteins. So this is the general structure of the amino acid. So there is also a side chain in the amino acid that makes the amino acid unique from other amino acids. So that's the beauty of the amino acids, because in the side chains there are many different R groups that are attached to it. Okay, can be classified based on the R group. Okay, so the glycine, for example, is the simplest amino acid in which the side chain is the hydrogen. It's just an H. So madaling ma-remember ang glycine na group. So it is also a neutral carbon because it's an inert carbon because it is H, yung basic din ito siya, uh, non-polar amino acids. So okay, it is unique because glycine, it is a chiral carbon because it will not that there is no stereoicity because there is there are two hydrogens that are attached to the chiral carbon. To illustrate or to give you a um, illustration about the amino acid structures, in which I know that you are familiar with it already because you are now taking BTEC 111, in which protein and it is entitled proteins and nucleic acids. So um, I'm sure that you already understood these concepts. So again, this is an amino group. It is composed of nitrogen, and two attached to it are the two hydrogen elements. And then there is also a carboxyl group that denotes that this is an acid structure. Okay, this is a carboxyl group. So yeah, so di ba ang sa acetic acid, so ang iyang functional chain or the side chain is a carboxyl group. Okay, so there's also the carbon that is attached to hydrogen, and there is the side chain that makes it unique from other amino acids. So that's the um, and this is another form of the structure: the carboxyl group, the H group, and then the amino group and the side chain. So this is the classification of amino acids. So there are four classifications based on the polarity, hydrophobicity, and the charge. Okay, so first classification is the nonpolar hydrophobic neutral, which is uncharged. Okay, another classification is the polar hydrophilic acidic, which is negatively charged. When we say acidic, it is really negative charge. And then another classification is the polar hydrophilic neutral, which is uncharged. Okay, like the glycine, right? So it is, yeah, it is polar pala hydrogen. Okay, the glycine is a polar hydrophilic neutral uncharged. So okay, and then there is also the fourth classification is the polar hydrophilic basic, which is positively charged. So these are the essential amino acids. So what do you mean by these essential amino acids? So these are non-essential. These are needed really by the body. Okay, so sorry for that. So essential amino acids are the methionine, threonine, histidine, valine, phenylalanine, isoleucine, tryptophan, lysine, and leucine. Okay, so for the peptide bond formation, how does the amino acids are attached together to form a polypeptide. Okay, so there is now the peptide bond formation, and the reaction is through the condensation reaction. Okay, it is between the amino group of the N residue and then also the carboxylic group of the N plus 1 residue. As you can see here that there is a peptide bond formation, and there is with this reaction, there is a loss of one water molecule. So in here, the amino group, so in order to attach, in order to attach one amino acid to another, so there is a loss of the what? Of the carbox of the oxygen, okay, of one oxygen there, okay, and then there's also a another loss of the one another amino acid from an loss of two hydrogen from another amino acid, and that forms an H2O. So with that loss of water, there will be now a peptide linkage for the two amino acids. So in order to also attach another amino acid, so a condensation reaction is will then be achieved, or will then be take place. Okay, so for the question that I was asking kanina, when can we say that we'll call it a peptide or a protein? So there is a rule there. So this is a linear arrangement of an amino acid residues linked by peptide bonds. So if the amino acids is um, below or below than 25 amino acids, okay, below 25 amino acids, okay, it is just generally termed as a protein. But if it otherwise, or the amino acids are above 25 chains or 25 acids, so it is termed as a protein. Okay? Peptides had directionality, like the DNA. Diba? We have directionality in the DNA, which is 5 prime to 3 prime direction. So here also in the peptide chains, there is a directionality in which there's an N terminal and the C terminal. Okay? So with that, there are hierarchy of protein structures. Depends upon the function of the proteins, and as I have remembered also in my BTech 101, in which I have also took that course when I was undergrad. So I have fun in learning many types of proteins. There are structural proteins, chaperone proteins, and many more. So there are also many types of structures of protein based on the function. There are four levels of hierarchy in which it is composed of a primary, secondary, tertiary, and quaternary structure. Okay, the function depends on the structure, right? Okay, so first hierarchy of the structure of proteins is the primary structure. Okay, so this is a linear sequence of amino acids from the N terminus, the amino group, to the C terminus, which is the carboxylic connected by a peptide band. So this is just a linear sequence. There is no other linkages to other amino acids on the middle and the end. So it's just a quite linear amino acids. And I think this peptide is a insulin. Okay, so mostly, so there are many also structures of it. Really is composed of primary structures of amino acids. No, so they're like that. So this is just a linear sequence. Mara siya kuan kanang beads, a string line of beads. Okay, so it's just simple, the primary structure. How about the secondary structure? So, okay, the secondary structure is the adjacent amino acids interact with each other, forming a repetitive conformation. As you can see here, class, that the secondary structure can form a alpha helix or the beta pleated sheet. Okay, here again, so in the secondary structure of a protein, there is a interaction of the amino acids between each of the amino acids that will form a alpha helix. So as you can see here, class, right now, so di ba kani ang iyang conformation based on the amino acid sequences. That's the linear sequence. Now, because of the different charge and the polarity of the amino acids, they tend to interact with each other, the adjacent amino acids. That is why um, there is a bond. There is a bond that will interact with each other. So that's it. There is a formation of bond, or there's a link between the oxygen and the hydrogen here in the adjacent amino acids that forms an alpha helix. While here in the beta plated sheets, there also here it is also different primary structures, right? Different chains of amino acids, and with this there is hydrogen bonds that form in the adjacent, which is the H and O in the adjacent linear sequence of amino acid, and it forms a beta pleated sheets, right? So there are it is brought about by the H hydrogen bonding of the carboxylic group, okay, the carboxylic group and the amino group. It will form a alpha helix and the beta plated sheets, and then there is also the beta bands. Okay, so there it is. Okay, another formation for the secondary structure. As you can see, you can form an alpha helix and the beta plated sheets, and there is an electrostatic attraction that forms a hydrogen bonding. It can be less electrostatic attraction. There could be hydrophobic interaction, and there is also the metal ion coordination, like in the hemoglobin. So there is a central ion there that will coordinate. That is why the iron is really important for the blood, the erythrocytes or the red blood cells, because it will hold the many amino acid structure to uh, functionally activate the in order to fulfill the role of the proteins in the blood. Okay, because the iron will also interact to the oxygen, so it is carrier of the oxygen for the distribution of the blood throughout the body. Okay, for the tertiary structure class, so this is a 3D structure as a result of the attraction of the R groups of amino acids widely separated in the well separated in the protein chains. It can be the hydrogen bonding, it could be disulfide bonds, the hydrophobic interaction, and then the electrostatic interaction. So let's just know this is the hydrogen bond, the interaction between the amino group and the carboxylic group, in which there is a connection between the hydrogen and the oxygen. Here is also the hydrophobic interaction. It is the clustering of the hydrophobic groups away from the water. Okay, the van der Waals interaction. But this is the polypeptide backbone, so the primary structure. While here, there is all an interaction of the sulfur, the two sulfur. There are amino acids that has a side chain involved, like tryptophan and cysteine, I think. Yeah, so that's uh, those are amino acids that has a side chain of sulfur, so it will then create a disulfide bridge. So one perfect example for that is our hair. Yeah, so those mga kulot og hair has many disulfide bridge, and because di ba our hair is composed of keratin. It is a structural protein. So keratin siya class sa atong hair. And then kung kulot ang atong hair, so there are many disulfide bridge. And then kay gusto man me, for example, I'm those who are fond of having a hairy bonding every year, because I want my hair to be straightened and not just to have more effort in doing my hair arrangement like that. And also my hair is not that good, it's not that presentable, so medyo wavy and very, so I opt do a hairy bonding. But really I have a natural wavy hair, so I have a many disulfide bridge in my hair, yeah, in the keratin structure. So that is why, so in the hairy bonding, so this disulfide bridge will then be dissolved. So that is why there is now the result of the hair is really very straight. So mechanical and chemical siya na uh, mechanical and chemical siya na factors, because di ba in the hairy bonding you have to apply chemical in order to dissolve the sulfide bridge. And then also they, after washing the hair and then also to blow it dry, they have to straighten it out using a hair iron. So it is in the denaturation of protein, there is a factor there that you have to subject it to heat, so that there will be destruction of the bridges, so the disulfide bridges. So that's a perfect example for the tertiary structure of the keratin protein in your hair. Okay, after that is there is also an ionic bond, which is positively charged ions, and then so negative charge that are interacting. So it also pertains to shape, which dictates the solubility in water. So this is now the tertiary structure. So there are many more interaction there already in the tertiary structure. Okay, so there are many examples for types of the tertiary structure. One is it is the fibrous proteins. The polypeptides are arranged side by side in a long filaments. Okay, yeah, and then also it's mechanically strong, stabilized by intermolecular bonding, and it is also insoluble in water. So have a structural or protective role, like the keratins in our hair. So it gives strength and rigidity to different organs of the body. So, and for example of it is a keratin. And also you have this collagen. So collagen is also famous because of the what di ba maraming ano ngayon, like the Bella Group um Bella Group company, that they sell collagen collagen, and also it is also famous in Japan that there are many products, skincare products, and also mga supplementary medicines that you can just really take collagen for your for the rigidity of your skin, so it prevents aging, diba? So that is fit for so that our skin will not sag. So that is why collagen is highly recommended for the rigidity or the freshness of our skin in our face, or also in our and the different parts of the skin. So that's it for the collagen. Okay. Okay, so next. So there are also globular proteins. It is a spherical shape here in the tertiary structure. So you'll now have this shape of the protein. So it will now determines its solubility. Okay, it is not really soluble in water anymore. And then the regular structural elements are mixed with randomly extended chain segments, and it is soluble in water. The amino acids in surface are polar, okay, and the inside is non-polar. Okay, so there are also hydrophobic patch, which is non-polar amino acids on the surface, and it is mobile with itself, like chaperone proteins. So there, it also belongs to the enzymes are one of those globular proteins. Enzymes have low activity without water. Okay. So there are also hormones that are made up of proteins, and then the oxygen binding proteins, which is the hemoglobin, right? So there are the iron, the oxygen will attach to the iron. So in the hemoglobin, the myoglobin, right, and the cytochrome. So that's those are examples of the globular proteins. Now, for the last kind of structure of proteins, it's the quaternary structure. Okay, so you also have this interaction between two or more polypeptide chains. So that's the one of the characteristics of the quaternary structure. It gives the biological activity. So, like the enzymes, like the hemoglobin, the role of the hemoglobin in the red blood cells, and also there is association of the tertiary structure. So, the hemoglobin, as you can see, that there are four different amino acid. Chains of amino acids, and it is quaternary structure, and there is an interaction between the polypeptide chains, and there are many there are higher iron or hem that are present in the different polypeptide chains. So that's the hemoglobin. Like this is the alpha chain, the beta chain, and so on and so forth. Okay, so the one polypeptide is a monomer, and then also two polypeptides is a dimer, a quaternary, and then oligomer, which is two or four. So that's the last part of the hierarchy of the protein structure. So again, the summary is that we have this primary structure, which is a linear chain of amino acids. So we call peptide is when it is less than 25 amino acids, and call it proteins is if it is more than 25 amino acids. Okay, and then another is that we have the secondary structure, that there is interaction between the adjacent amino acids that will form a beta sheets, and also this alpha helix. And then tertiary structure is the two or more interaction between the different amino acids Okay, different interactions. Could be disulfide bridges, could be electrostatic interaction in the hydrogen bonds. While in the quaternary structure, there are two or more polypeptide chains that are interacting with one another, like the hemoglobin and then the collagen. Okay, so let us go now to the protein denaturation. How does the protein is denatured? So, what's the characteristics of the denaturation of the protein? So, by definition, it is the modification in the secondary, tertiary, and or quaternary structure of proteins without the rupture of the polypeptide bonds. It's bond, yeah. So it could be the modification of the secondary structure, tertiary, and the quaternary structure. Okay, it can be a reversible or irreversible change of native conformation. So when there is reversible change, it could be like that, can be reconstructed. But if it is irreversible, so there should no the denaturation, the protein will not be used anymore. Like for example, if it is enzymes. Okay, so what are the changes you see during the denaturation? Okay, so what are the characteristics when the proteins are denatured? There is a decrease of solubility in water, so it will not be dissolved anymore. So there, the non-polar amino acids are exposed. Like for example, yeah, so it will then be discussed in the exercise 4 and 5 because it is relevant here that the milk proteins are not will be precipitated and will not be soluble when there is addition of acids. Okay, so it can also be susceptible to enzyme attack because of the exposure of the polypeptide bonds and the cleavage of bonds. And the third is the biological activity or function is lost. So meaning it will not then, if it's an enzyme, so it will not be used anymore for such activity or the for example production of or the degradation of such carbohydrates. Yeah, so that's it for the protein denaturation. So what are the agents of the protein denaturation? So what are the factors that these proteins can be denatured? One example, one factor is heat. Diba? So I think we can observe this in a daily basis because I know we are at home and we are always cooking, right? So all interaction can be destroyed through heat. And for perfect example is when we cook egg, diba? As you can see, kung hindi pa siya luto, so it's transparent, right? So, pag siya slimy and it's transparent, ang egg white niya. But if it's already cooked, so it's now insoluble. So there's now an egg white. It's very white, right? So there's also another agent is the organic solvents, which is the chloro-2-ethanol, which is non-polar hydrophobic acid. In which one perfect example of this class is that when we want to extract DNA, so particularly in a bacterial cell, so you have to, or in the plant cells, you have to use such strong solvents because they are really these are strong solvents. These are non-polar hydrophobic acids that will really denature proteins and one organic solvent that are commonly used in the DNA extraction is the chloroform, so that there will be denaturation of those proteins and other fats. Okay, so um, particularly or specifically, it causes change in the polarity of the solvent, and there will be exposure of the non-polar amino acids. So another factor or another agent is the extreme pH. So at neutral pH class, the most proteins are negatively charged and few are positively charged, right? Because of the different side chains of the amino acids, right? But the pH, the net electrostatic repulsive energy is small, thus proteins are more stable. This is a neutral pH. But if there is now the isoelectric point, this is the isoelectric point. At extreme pH values, there is strong intermolecular electrostatic repulsion due to the high net charge results uh, when we say that uh, we add we add acetic acid, so there is now a highly negative charge. So there is now a high net charge results that will result in swelling and the unfolding of the protein molecule. As protein is really a pH sensitive because of the side chains of the amino acids. So that is why in the in if you are dealing with enzymes, so you have to maintain, or you have to take care of your enzymes, that you have to maintain a buffer. So when we say that we are doing when we're doing fermentation, enzyme fermentation, dili siya bak ka ng microbial fermentation, na enzyme fermentation siya. So naadyo siya pH i-maintain, because if there is a change of the pH, increase the change of pH, or the decreased change of pH. So there is a possibility that your enzymes will then be denatured. So the result is that there is swelling and folding of the protein molecules. So another agent of denaturation is the presence of organic compounds, or the urea, and then the amino group is capable of forming hydrogen bonds with amino acids of proteins, and then hydrogen bonding in between amino acids is destroyed, and the organic compound is capable of forming hydrogen bonds with water. There is a change in polarity with solvent, and hydrophobic interaction is destroyed. Okay, so there is also a presence of acid or base, extreme pH. Okay, basic pH or the acidic pH. There's also the presence of alcohol, like the SARS-CoV-2, diba? We are always disinfecting our hands so that the SARS-CoV-2 will then be, the spike protein of the SARS-CoV-2 will be denatured. Well, yeah, will be denatured, and that's the agent for the denaturation of the protein. We have also here the presence of reducing agents, okay, like for cysteine, yeah, and then there is a um, the the separation of two cysteines in the presence of the reducing agents. So I guess that ends the lecture about the discussion for the exercises 4 and 5. So I'll pause for a while, class, so that you can take a break. Okay. Okay, class, I'm back again. So for the next slide, okay, this is now the exercise 4. It is entitled Protein Purification by Acetone Precipitation. Okay, so I have already discussed with you the different agents of denaturation. It's relevant here that the solubility of proteins is dependent on, among other things, the dielectric constant of the solutions. So the water and the dimethyl sulfoxide can stabilize the interaction of the proteins between themselves and protein molecules and favor the dissolution of protein. So organic solvents like acetone and methanol discourage the dispersion of protein molecules in the media. So with that, the solubility of the proteins can be lowered, as when we say protein denaturation, one of the characteristics of the protein denaturation is that there is lowering of the solubility of proteins, and precipitation can be induced by lowering the effective dielectric constant of the meat. Okay, so it is accomplished by adding water-soluble solvents such as acetone to an aqueous solution of protein. So it has the advantage that it is relatively cheap and available in a pure form with few contaminants that may inhibit or poison the enzyme. Okay, for the objectives or the learning outcomes, so objectives for the lab report, you have to specify, put their objectives, okay, to recover proteins or enzymes from a solution by adding acetone. Okay, these are the materials that you have to gather if you are in a laboratory setting. So, but unfortunately, we cannot do it in the lab, but we will do it. You will do it at home. Okay, so this is the home setting experiment. You have to buy because this is acetone is really available in the market because it is used for the cosmetic purposes. Okay, you have to buy 20 ml acetone or a larger one. You have to obtain also powdered milk, any brand that are available at home. So could be 1/2 tablespoon of powdered milk, and you have to take tablespoon baby dropper also. Then maybe you can ask from your neighbor if they have babies. You have to ask it, but don't return it anymore. Okay, so you have to discard it now. So you have to also obtain a clear glass, and it must not be used after experiment, and also warm water. Okay, so these are the procedure for this is the procedure for the experiment at home. You have to dissolve, actually not inward 5 grams but 2.5 grams lang. So take note of this plus 2.5 grams of powdered milk with approximately 20 ml warm water. Okay, using a dropper. Okay, slowly drop acetone into the milk while stirring. Okay, and then continue add acetone until the milk becomes clear and no protein is separate. Okay, by the way guys, 2.5 grams and then 10 ml na siya ato siya pagamay ang iyang volume. Okay, 2.5 grams powdered milk and then 10 ml warm water. Okay, and then after that you have to estimate the volume of the acetone added in the milk using the calibration in the dropper used. Okay, there will be an estimation of the volume. Make sure that the dropper has a calibration, diba? Ang sa dropper, mga 2 ml or 1 ml naman siguro na. So, you have to estimate the volume added, the acetone added in the milk, okay? And then you have to conduct it in two replications. Take a picture of your mixture before and after, okay? And please include it in your discussion. So watch the additional resources in YouTube for additional insights and understanding. So that's for the exercises, exercise 4. So this, I won't discuss to you the laboratory setting because I have to, I know. So this is the data to be analyzed. Okay, so this is a presumed data. Okay, so this is the presumed data that you have to also to include in the discussion. Also, yeah, calculate the weight of the protein assuming you obtain the data of the following. So replication 1, 2, and 3. Okay, you have to insert this. And these are the questions that you have to answer. Okay, number 1 is to state the principle why there is fast separation conducted in lower temperatures. And second is what is the difference of the composition of powdered milk and the pasteurized milk. Which of the two yield more milk proteins? Okay, so this is now the instruction on how to prepare and submit the laboratory report. I won't dwell on this for too long because I know that you are already making lab reports right now. So please don't forget to uh, another parts of your lab report. Okay, because every part of your lab report has a corresponding point. Okay, so for the exercise 5 class, so this is naman protein purification by isoelectric precipitation. So it's just the solubility of the proteins depends also on the pH of the solution. As what I have also discussed kanina, that one of the agents of denaturation is the extreme pH. So the protein can either have positively or negatively charged overall due to the terminal amino acids and the carboxyl groups and the groups on the side chain. Okay, so it is positively charged at low pH and then negatively charged at high pH. Okay, meanwhile, the intermediate pH at which a protein molecule has a net charge of zero, called the isoelectric bond. Now, generally, the net charge in the protein, either positive or negative, can interact with molecules. It is likely that protein molecule to dissociate itself from other protein molecules. Thus, it will become more soluble. So, protein is the least soluble when the pH of the solution is at its isoelectric point. So, like for example, when microorganisms go in milk, they often produce acids and lower the pH of the milk. Like what? Like the yogurt. So what? That's one perfect example is that, diba, ang Streptococcus bulgaricus, mga microorganisms, so tend to lower the pH and it will make the milk um, sour, and also make the milk uh, viscous and stable. No? So the phenomenon is precipitation or coagulation of milk protein, which is the casein at low pH as milk becomes spoiled, is one of the common examples of protein isolation due to changes in the pH. Okay, that this is yogurt. Okay, so the objective of this exercise is to recover protein enzyme from a solution by changing the pH of the solution. Okay, so I guess this is now the experiment at home method. So you just have to obtain 5 grams powdered milk, any brand, and 10 ml vinegar, just any brand also, and then water used dropper, and then the wood sticker and clear glass. So the method is just simple. So you have to dissolve approximately 2 tablespoons of powdered milk into a 10 ml warm water in a clear glass. Then let it cool. Then slowly drop vinegar using dropper into the milk with constant stirring until precipitate is formed. Continue add vinegar until there is the mixture of clear and no precipitate is separated anymore. You have to estimate the volume of the vinegar added using the dropper if there is calibration. So make sure that your dropper has calibration and conduct into replications. And the last thing you have to do is that you have to take a picture of your mixture and then include it in your discussion. So that's it. These are the questions you have to answer included in the lab report. So, what is the typical pH of the powdered milk when it is dissolved in the water? Okay. Explain how it affects the protein separation to the pH of the milk, and compare the yield of the milk protein from the previous experiment. That is why I have to give it to you. uh, I have to give the exercise 4 and 5 na. Okay, so what are the advantages of and the disadvantages of using acids for the precipitation of proteins? So, okay, if you have time, please watch this video so you can have more insights for the exercise 5. Okay, so I think that's That's the last slide, and that ends the laboratory part of the INBT 121. So thank you so much for listening. And before I end here, I would like to encourage you in Psalm 121, 1 to 2. It is a Bible verse that I lift my eyes to the mountains. Where does my help come from? My help comes from the Lord, the maker of heaven and earth. So you have to lift up your eyes to the mountains or to God. So our help really comes from the Lord. So whatever things that you are burdened with, and the things that you have to do, and if you are overwhelmed by many circumstances, not just the academic requirements but also personal problems, so you have to ask the Lord and you have to trust the Lord, because know that He is our help and He will always be there anytime. So it really pays that we have a relationship with the Lord Jesus Christ all the time. So it will not just guard, or it will just help us in our for the um, uh, easing our burdens or our problems, but it will really make your it make it will make our life worth living, and also make our life more secure, and also it will guard our hearts and our minds, so it will really have a relevant it will have a great impact to our mental health, because when we di ba, we always have to think of our mental health when we are overwhelmed too much, and we have so much to do, and we might end up sad and depressed and many other things. But it's quite natural that we are really having those emotions, but when we really not shrink in that moment and will not just stuck in there. So we have to go to the Lord and ask for help, knowing that we cannot do things on our own in our ways. So it also humbles us to really lift our eyes to the Lord and acknowledge Him that He is our Lord and He is the Savior. He is our help. So that's it for our mental health, because diba, so October was a mental health month, but it should be every day that we are always having this encouragement, mental health encouragement. But I rather prefer to really encourage myself with spiritual things, because when we know that we really come from God and we are really designed because of the Lord, our purpose is because of the Lord, so we will have a direction. We are sure of ourselves, of who we are, our identity, and our drive to continue all the things that we are going to do. Okay, so thank you so much, class, for listening once again, and I hope that you have a good time watching this video, and I hope that you will also um, um, prepare now the materials for your experiments. I will give you 3 weeks, okay, for this experiment, so it can be done on the same setting na for exercise 4 and 5. Okay, but separate ang ilang lab reports. Okay, separate na siya. Okay, we'll just post in PSUEE the submission for these exercises 4 and 5. Okay, thank you so much, class, for listening, and have a good day. Goodbye.